So thank you, David, and thanks to Sophie for inviting me here to talk today. And so the title of my talk is a bit vague because I wanted to cover several different things rather than focus on one particular ion channel, but rather a few different ion channels that we're working on and some of the modulation of those ion channels. So obviously in this audience people are working on different things, so I'll give a very brief introduction to nociception, seeing as I'm the first one, I think, of the few people talking about it. So pain is defined as an unpleasant sensory and emotional experience associated with or resembling that associated with actual or potential tissue damage. But in our lab we primarily work with animals that are non-human, and so it's very difficult to interpret the emotions of a mouse no matter what we might think. So although I'll use the term pain, what we're really measuring in the lab is nociception. So that's the neural process of encoding noxious stimuli. So we're interested in the lab about how nerves are switched on, how that process changes from health and disease. And of course, the context of this conference, the ion channels involved in that control. Why is this something we should be doing? Because, of course, when you talk about working on pain and working with animals, that raises lots of ethical concerns. Well, if we look at the prevalence of chronic pain, this is looking at lots of different studies. When you average them out, in the UK, we've got about 43% of the adult population experience chronic pain. And that's a lot of people. And of course we've got painkillers, but they don't always work. So if you ask people who experience chronic pain whether their pain medication is adequate, only about 1/3 will say it is. So we have problems of side effects and problems of a lack of efficacy. And the other thing we need to be concerned about is that this problem is going to get a lot worse, because if you look at the prevalence of chronic pain with age, as you get older, the chance of you having chronic pain increases. Hopefully we won't have increased problems of the connectivity. But anyway, so this is a big problem. We've got an ageing population, the drugs don't work, we need to understand more about nociception to develop new ways of combating it. So for a bit of basic anatomy, there are two main types of sensory neurons. We've got the nociceptors shown in red. Those are the exciting ones because they're the ones involved in the transmission of noxious stimuli that make you go ouch. The mechanoreceptors are important, but not for the purposes of this talk. They're the neurons that are involved in the sense of touch. So all of these neurons innervate an end organ, in this case the skin, and when activated an action potential will fly off to the spinal cord, and that's the first synapse. So these nerves can be very long, from your little toe to your spinal cord, probably some of the biggest cells in your body. The cell bodies for these are located for everything below your head in the dorsal root ganglia, the DRG, and so we can isolate these and culture them and use those as a model for what's happening at the nerve ending, because to study ion channel behaviour when it's embedded in lots of other things, it is complicated. So in some of the data I'll be showing you, it will be from these DRG neurons, but not all of it, because the problem with DRGs is neurons can respond to lots of things. If you're trying to study one ion channel, you obviously need to have that in a bit more of a focused environment. And that's the case when I'm going to be talking here about modulators of the acid-sensing ion channel ASIC3. So why are acid senses important? Well, tissue acidosis occurs in many painful conditions. So you've got osteoarthritis, rheumatoid arthritis, you get a shift in the synovial fluid down to sort of 6 and a bit, but also lots of lung conditions. So cystic fibrosis, you get a shift from 7.3 to 7.1. And of course, when we get an acid shift in these conditions, that's not the only parameter that's changing. So it's important to bear in mind that ASIC3, for example, is modulated by lots of other substances associated with inflammation, such as arachidonic acid. So even though these pH shifts may not seem that extreme, pH will work in concert with other mediators to activate these ion channels. Working with protons is horrible, because the body has evolved lots of ways to detect them. So we've got the acid-sensing ion channels over here, not all of whom do what they should do, because ASIC2B and ASIC4 don't actually sense protons. If you make them, they do form heterotrimers, and then they will modulate the function of the ion channels, but as homomers, they're not proton sensitive. There are lots of TRP channels that can be modulated in different ways, TRPV1 probably being the most famous, but activated at a lower pH than some of the ASICs. The K2Ps can be modulated, or some of them can be modulated by low pH, and then the largely ignored family, the proton-sensing GPCRs over here, and I'll come back to those in the last part of my talk. So why is ASIC3 of interest? Well, there's two main reasons. First, if you look at the expression of the different ASIC isoforms in the mouse at least, it's the most heavily expressed one in the dorsal root ganglia, so those neurons that are going to be sensing acid as a noxious stimulus in the periphery. The other thing is that when you activate an ASIC under normal conditions, it has a two-phase response. It's transient current, which is massive, but it rapidly inactivates. ASIC3 is unusual because it maintains this sustained inward current in the presence of low pH. So that means even if we ignore this bit, because it's going to be over very quickly, when we have a sustained low pH environment, ASIC3 will be depolarizing that neuron. So pH is an important activator of these, but about a decade ago now it became clear that they could be activated by other things. So this group identified there is a non-proton ligand sensor on ASIC3. And they demonstrated that this molecule, GMQ, could potentiate the pH7-induced current, But perhaps more importantly, it could also activate the ion channel at neutral pH. So this is the pH response, and now we've got GMQ in a dose-dependent way activating the ion channel. So we teamed up with Tofik Rahman, who's in the audience here, to ask if we could use GMQ to guide an in silico screening of FDA-approved drugs to see if anything that's already out there might have some side effects or therapeutic effects through modulation of ASIC3. So Tofik led all the in silico work, and then Jared Cahilio was a postdoc in the lab who did all the electrophysiology. So any in silico questions, and hopefully Tofik can take those, but briefly what we've got here is that these are our top 5 hits that we came out with. And we've got 2 different scores, the shape Tanimoto, so this is telling about the 3D shape similarity, and the colour Tanimoto, which is the one its chemical features. So GMQ has its guanidine group, which is also present in guanabens, and all of them have either an explicit or an implicit guanidine group. So what do these molecules do? We found them through an in silico screen. Is that just by luck? So we're looking here at the pH7 induced response of ASIC3, and this is GMQ as our positive control, and others have shown it potentiates that response. And essentially 3 out of our 5 potential hits produce the same sort of effect. So tizanidine, bromidine, and guanabens cause this increase in the peak current density, which is summarized here. So that's nice, they can potentiate, but what about their ability to activate? So we looked at that in a slightly different way. We're looking at the modulation of the pH6 induced current. So what we've got here is APETX2 is a well-characterized ASIC3 inhibitor which inhibits the current. And essentially none of these compounds were observed to produce a current at neutral pH or modulate the pH6 current, apart from guanabens. You can see here it increases the peak current at pH6, which even GMQ doesn't do, but more importantly, it also activates the ion channel at neutral pH, just like GMQ. So that's what you can see here. We've got the pH-induced current. When you apply GMQ, it's not smaller, but sustained current, and we see the same effect with guanabens. So I'm not going to show you all the in silico modelling, there'll be a bit at the end, but essentially these molecules appear to bind to the same non-proton ligand sensor domain of the ion channel. So guanabens is used therapeutically as an alpha-2 adrenoceptor agonist. It's used as an antihypertensive, not very frequently used, but it can be. And a derivative called cephin-1 does not. So this is the guanabens, this is cephin-1, and essentially the only difference is we've lost a chlorine atom here. So why is cephin-1 of interest? Well, it's been proposed by another group to inhibit this regulatory subunit of protein phosphatase, this thing here, and it has a role in being used as a target for treating protein misfolding diseases. So we were just wondering if this compound that's going through potentially into the clinic, whether it might have any effects on ASIC3, which we'll find out on the next slide, hopefully. So, essentially, yes, it does. It does exactly what guanabens does. We've got our pH6 induced current, and cephin-1 is also able to induce these sustained currents. Notice, though, this is at 1 millimolar, so this is not particularly potent. If we compare the effects of GMQ, guanabens, and cephin-1, you can see that GMQ gives the biggest response, so we can say it's perhaps more efficacious, but the guanabens is slightly more potent. We can also see that cephin-1 potentiates the ASIC3 acid activation in the same way that GMQ did, and again you get this potentiation of both the transient phase and the sustained phase. So when we do look at how these molecules interact with ASIC3, at least from in silico predictions, you're looking here at the non-proton sensing ligand binding domain, and this is 5 different poses of how the drugs are thought to interact with the ion channel, and they're all binding to approximately the same state. So we haven't gone through and done site-directed mutagenesis to prove this, because some of the problems are that when you mutate the residues involved, you end up with a non-functional ion channel, which is a very easy way of avoiding doing experiments, but it's just true, unfortunately. So we don't know for sure, but that's the prediction. We've obviously got some slight differences in how these compounds regulate the ion channels, which probably are accounted for the fact they do have slightly different chemical interactions and therefore will not interact with the ion channel in exactly the same way. But essentially what this shows is that we can use in silico docking to look at molecules that are already out there for other effects they may have. And we've got ongoing work looking at other molecules that inhibit the ion channel rather than cause its activation. So that's what I want to say about how we can transduce a nociceptor. If we move on to look at transmission, because obviously there's 2 main sorts of ion channels we need to consider here, voltage-gated sodium channels and voltage-gated potassium channels. So hopefully everyone in this audience knows we've got these rather large alpha subunits, but then we've also got the much understudied beta subunits which regulate these alphas. So this is just a very short amount of data to essentially say, please go and look at Alex Cloke's poster upstairs. We're trying to look at how the beta 3 subunit in particular can modulate NAV1.7. And so we've got a few different tools we can use for this. We can use adrenaline-Cree. So adrenaline-Cree is, adrenaline is only expressed in sensory neurons. This enables us to delete NAV1.7 just in DRG neurons, and we can look at how TTX gets rid of all the other things, and we can identify our TTX sensitive current in both the wild type and the NAV1.7 knockout and infer the contribution of 1.7. But also there are very good toxins that enable you to identify the role of 1.7. So Protox 2 is an inhibitor of 1.7, and again, by applying this and doing the subtraction, we can work out what the NAV1.7 contribution is. So that's good for being able to look at the alpha subunits, but the problem is, how do you work out how the betas are modulating, because there are no drugs to modulate beta subunits. So we can't do it directly, but what we can do is use these tools to say what is going on in our neurons with 1.7, and then have a way of identifying which beta subunits are there. So this is early days, but essentially what Alex does is he harvests the cytoplasm after he's made his recordings, you then reverse transcribe things, and you do a nested PCR to detect the gene fragments to say, okay, in the neuron I've recorded from, what sort of 1.7 current did it have, and how then could that be modulated by different beta subunits present? So you end up with just amplifying up your very small product. There are lots of things you have to consider which are annoying if you're an electrophysiologist, because you want to get on with your experiments, but here you need to have very RNase-free solutions. You have to autoclave and sterilize all sorts of things. It's a bit annoying, but you do get some quite nice data. So this is still at preliminary stages, but this is just showing that from the 6 cells recorded from here, they've all got 1.7, and at least 5 of them have the beta 3 subunit. So the aim is to use this platform of using genetics and pharmacology to identify what neurons have 1.7, and then in a post hoc manner be able to work out, okay, and was the beta subunit there? What happens if we knock down the beta 3? And the reason we're interested in beta 3 is the knockout animal itself has an analgesic phenotype in certain inflammatory pain studies. So that's very much just a shout out to go and talk to Alex for more details. So voltage-gated sodium channels are important, but also we need to think about how we can get things repolarized. And that's what comes up here with this tale of painless delivery, because we're going to talk now about voltage-gated potassium channels. So there are genes we know that cause congenital insensitivity to pain, and there are genes that can cause extreme pain phenotypes. So this is a very large collaboration led by Jeff Woods and Mike Lee up at Addenbrooke. It's been involved many other people, and we wanted to see if we could identify new genetic pain variants by studying painless labour. So there are certain people who, when they give birth, have no requests for analgesics. So we researched people who had no reported pain, no requirement or request for analgesics, and were otherwise healthy. Once we had those people, we then asked them to come back to the clinic and undergo quantitative sensory testing to see, is there anything else unusual about their pain phenotype? So we're looking here at their warmth and cold and pressure detection threshold. No real difference, there's a slight change in the warmth detection. When we look at cold pain, heat pain and pressure pain, these women on the right, shown in black and red, these are our people who did not request analgesia during childbirth, and they have a high pain threshold regardless of the stimulus. So the 3 red triangles are the 3 people I want to talk about, because they all have the same genetic variation. And that genetic variation is in KCNG4, so it's overrepresented if we compare that to 1,000 genomes. So we essentially had 4 heterozygotes, and that's important, compared to an expected about 1. So we've had this gene and thought, hurrah, we've got a thing we can study, and then got a bit disappointed because KV6.4 is what's called a silent subunit. So you have to study how it modifies other things. The mutation itself is in the selectivity filter, and unsurprisingly, the mutation is then very well conserved. The lack of mutation is conserved in lots of other potassium channels. And so 6.4 is known to modulate biophysical properties, so presumably our genetic variation here must be causing a change in that modulation. So we started off in HEC cells, we're measuring the activity of 2.1 homers, and we're looking at the inactivation. So around the resting membrane potential, everything's active. Throw in the wild type 6.4, and that's a very large change. 6.4's job is essentially to regulate 2.1, so that around the resting membrane potential, a lot more of the ion channel is not functional. So what happens then with our rare variants that are identified through studying these people with a high pain threshold? Well, it essentially doesn't do anything. It goes back to where you started. So this is a massive shift, and when you have this variant, you lose what's going on. But here we're just co-transfecting into HEC cells, and we have to remember that all those women were heterozygotes. So if we just look at what this shift in the V half is, these are the important group, because now we've transfected in the common and the rare variation, and you have the same phenotype in terms of the V half for inactivation as when we just have the rare variant. So essentially this rare variant acts in a dominant negative fashion. So how is it doing this? Well, if we do some cell biology and we look at cells that are expressing these different variations of 2.1 with or without the different forms of 6.4, We've got the sodium-potassium ATPase as a marker of the plasma membrane. We can use a HA tag to examine where 6.4 is. And in the normal situation with the common variant, we can see that we get the 6.4 around the plasma membrane. So if you do a line scan through things, you get these green peaks overlapping with the red peaks. So the ion channel is where it needs to be to function. If we put in our rare variant, we do not get that. It just sticks in the middle of the cell. And importantly, if we have, again, the co-transfection, everything just stays in the middle. So we're not quite sure how this is happening, but this rare variant is preventing 6.4 getting to the membrane, which means you just have 2.1 sitting there doing its thing. So obviously this story started with labour pain, which is very difficult to study in rodents. There is one paper I'm aware of where someone has sketched out how the rodent moves differently when giving birth if you do or don't give it morphine, which didn't seem like a very objective way of doing things. So we just wanted to say, okay, well, is 6.4 where it would need to be to detect labour pain? So we conducted retrograde tracing of the uterine horns. You get some blue cells, and we just conducted single-cell qPCR to say, is it there? So KCNG4 is our 6.4. Yes, it is present in neurons that also express 2.1. Importantly, it's also in neurons that express TRPV1 and NAV1.8, which you could argue are markers of nociceptors. It is present elsewhere in the nervous system. I'm not trying to say this is the only thing that's going to be controlling nociception, but at least it is expressed in nociceptors. So a nociceptor, of course, is the important bit for turning your pain signal on rather than a hex cell. So we then looked at what would happen if we overexpress the common and rare variants in neurons. So we've just got a current ramp here. We're depolarizing the cell. The common form is 6.4, the rare form, and you can see we need a greater amount of current injected into the cell to drive an action potential firing. And that's summarized here. So you have a higher action potential threshold. So in cartoon format, what's going on then is in a normal situation you have 2.1 on its own, but you also have 2.1 being regulated by 6.4. And because of how it shifts the inactivation curve, we have less potassium leaking out the cell, and therefore we've got cells that are ready to be switched on. By contrast with our rare variant, you no longer have this. You end up with just 2.1 at the membrane, which has a very different inactivation threshold, and it's essentially leaking potassium around the resting membrane potential. So you need a much greater input to drive pain. And so this is the study we started off with, collaborating with Jeff, and then we last year were awarded a large pain consortium, which is called Advantage, which can be looking at all sorts of visceral pain. And part of this, looking at people with all sorts of things from pancreatic pain to inflammatory bowel disease, is going to be genotyping all of these people to hopefully identify even more common variants that might, or rare variants that might underpin pain sensation. So I want to finish in the last few minutes by bringing us back to proton sensing and not talking about neurons. But we need to think about neurons if we're going to drive pain. So if we look at a horribly complicated diagram of a rheumatoid arthritis joint and an osteoarthritis joint, there are lots of different cell types and lots of different mediators. An important cell type in both circumstances, though, are the synovial fibroblasts, because they're always there. We don't need to worry about things infiltrating in and spitting stuff out. The synovial fibroblasts are there from the beginning. As I've shown you before, lots of different proton sensors. We're going to talk now about proton sensing GPCRs. Specifically, GPR65, and this is all the work of Luke Patterson, a PhD student, now postdoc in the lab. If you put protons onto anything, you've got no idea what's being activated. So this compound, BTB, had been developed by others as being a selective GPR65 selective agonist. In stable cell lines, we can see that if we're recruiting cyclic AMP, it only activates GPR65. So Luke was going to use this as a tool for probing the role of GPR65 in pain sensation. So if you inject it into the knee of a mouse, you get swelling, which is a good start, unless you're the mouse. We get a decrease in the time spent digging, which we know is a behavioural output of having excitable knee-innervating neurons. We have no change in the ability to run on rotor rods. The animals aren't becoming lame. And also, if we squeeze the mouse knee and you look at the withdrawal threshold, we have a decrease in pressure. So BTB causes swelling and causes pain-related behaviours. So we thought, OK, well, what will BTB do to neurons? And the answer was not a lot. If you put it onto neurons, no change in the activity of the action potential threshold. We looked at other parameters, nothing changed. But the animal behaviour changed, so there might be some communication going on between neurons and non-neuronal cell types. So what Luke then did is he injected the animals with fast blue and then injected them with BTB. So we can look at those knee-innervating neurons and how they might be modulated. Same behavioural data as before. We get an increase in swelling, we get a decrease in pain thresholds. When we look at those fast blue labelled neurons from the animals that are injected with BTB and are in pain, we have this dramatic drop in the action potential threshold, which is presumably why the mice are in pain. So what other cell types might be involved? Well, as I said, the synovial fibroblasts are present, and we know they express GPR65. So what Luke did is he applied BTB to FLS cells, took that media, put it onto DRG, and we could replicate that effect. So it looks then as though BTB is activating the synovial fibroblasts. This in turn is spitting something out that is then modulating ion channels of which we don't know which, because lots control the action potential threshold, and therefore we end up with hyperexcitable neurons. Which mediators might be involved? We've carried out a cytokine array under different conditions, and essentially got numerous mediators, which is the next thing we need to work out, that are upregulated in FLS through activation of GPR65. So we need to work out which of these mediators are causing a change in the action potential threshold, and of course which ion channels are involved in that regulation. So to summarise, transduction and transmission are key to nociception. Current proposed therapeutics do actually do things to ASIC3 similar to GMQ. I think that shows the benefits of doing a in silico approach for studying some new things before just throwing stuff onto cells. We've got a method in place to determine how beta subunits may be regulating alphas, and I've shown you this idea by you study pain in the wild, you might identify genes that are involved in pain transmission and transduction. And I think 6.4 is something we're interested to see whether this is another potential target for analgesia. And again, there's more to nociception than nerves. We have to remember that mediators can affect other cells that release things onto the nerves. And just thank everyone who did the lab, everyone who did the work in the lab, those who did most of the work are shown in bold. Thanks everyone who's given us money, and thank you for your attention.
Very nice, thank you. We'll take a few questions. Is anybody? Yes, at the back, please. Give your name first, please.
Hi, I'm Alex from Meteor Biosciences, and just a general question, I might have missed it. So in your women with painless labor, so you mentioned obviously DKC and G4, the 3 recurrent mutations, but the other patients, did they also show in general mutations in the same channels? Or like, was there a pattern, or was it?
So we have those 3 red triangles, which are 3 women which had the KV6.4 variant. Of the black circle people, there are some other variants we're looking at, and there are others where we're really not sure what's going on, because obviously if you have a variant that might be explaining your phenotype, you need it to crop up more than once to think that's of interest. So we do have 2 other genes in those black circles that we're trying to work out if that variant that's common to those people does change nociceptor excitability. So watch this space.
Thanks.
If I could ask, what's the phenotype of these women? You've described one that they feel less pain in labour, but have they experienced other things about pain? No,
so none of them, before they were sort of found through a study, none of them had reported having particularly high pain thresholds or unusually sort of passages through life. So it's not like the people with congenital insensitivity to pain, which will often present with all sorts of injuries and other problems, where it's clear from early on something is different about their nociception. So some of them, when they went through the QST and were saying, look, you've got this high pain threshold across all these things, then said, oh, well, maybe that makes sense, but it's not high enough to really make them stand out as being particularly unusual.
Okay. Is anything known about their family histories, whether family members suffer from neuropathic pain less, or are there any connections with that?
I'm not aware we've got that data, is the honest answer.
Okay, that's very interesting cohort.
We've got another question here.
Yes.
Hi there, Martin Gunthorpe. If you take us all the way back to the ASIC3 part of the talk with the GMQ, so that was looking at modulators which are increasing ASIC3 function. Did you actually find any inhibitors which could shut down the channel? I guess that's therapeutically relevant as well.
Exactly, no. So no, we didn't with that study because GMQ was used as our bait, and GMQ is obviously known to activate. But there are papers from about 20 years ago showing that non-steroidal anti-inflammatory drugs inhibit ASICs. We've used a similar approach, and we've identified other molecules that do have a similar ability to inhibit ASICs, or ASIC3. So the same approach has been used, we just haven't published the inhibitory work yet. Because you're right, from a therapeutic point of view, that's potentially more interesting.
Yeah, and as a modulator site, you would imagine that you could get modulators which acted as negative allosteric modulators via the same binding opportunity as well. So,
yeah, so no, none of the GMQ related molecules, those there, they did nothing like 2 of the ones I showed, or they caused the same potentiation.
Thanks.
Yes, another question over there.
For the GMQ binding site, have you tested it's not allosteric to the proton binding site? Does it shift the pH dependence?
it does shift the pH dependence, but it isn't. The problem is with the proton binding site is also up for debate as to where it is, because the original crystal structure says there's this thumb domain that does this, and it's got these carboxylate pairs, and your ASIC2A, which is acid sensitive, doesn't have those carboxylate pairs. So I would argue we know there are lots of sites involved in proton activation, but it's more complex than just saying there's 3 carboxylates which get protonated. So I think we don't understand enough about proton sensing to understand how non-proton sensing integrates with that.
Right, but it's still feeding into the same mechanism. It's affecting gating through the.
Yes, because when GMQ causes this current, it's thought to be shifting you get the window current that's being activated by GMQ. That's the idea.
Good.
Anyone else?
I think we better finish there. We started a little bit late, but I think we're doing good for time. Thank you very much indeed, Ewan.